DNA replication is essential for cell division, cell growth, and cell repair. There are several proteins that the cell utilizes to ensure that this process is completed smoothly. It is important to remember that cells and their components are not conscious and that they, therefore, cannot want to undergo certain steps. Rather, it is important to remember that this entire process is dictated by an evolutionarily cultivated sequence of chemical interactions based on the properties of the individual components. The first step is the unwinding of the double helix. This is done by the enzyme DNA helicase. DNA helicase are motor proteins that move along the sugar phosphate backbone of the DNA molecule, annealing or separating the parent strand into two by breaking the hydrogen bonds that connect the complementary base pairs of each strand together. The replication fork is the Y-shaped area where the helicase has split apart the two strands of DNA. The single-stranded DNA, or ssDNA, is less stable than its original double-stranded form and is hypersensitive in nature. To protect the genome, single-stranded binding proteins stabilize the individual DNA strands. In this way, single-stranded binding proteins maintain the annealed structure of the single-stranded DNA. The enzyme RNA primase then synthesizes RNA primer, which is attached to the strands where the DNA synthesis needs to begin. DNA is made up of nucleic acids, which are further made up of a pentose 5-carbon sugar and a phosphate group. The carbons in the pentose sugar are numbered 1 through 5. The carbon numbered 3' with the hydroxyl group and 5' with the phosphate group are distinctly located at the ends of the DNA molecule. DNA is always synthesized in the 5' to 3' direction. The leading strand is synthesized continuously in the direction that the helicase unzips the DNA. The lagging strand is synthesized discontinuously, away from the direction that the helicase unzips the DNA. Because DNA is always synthesized in the 5' to 3' direction, the lagging strand is synthesized in fragments, known as Okazaki fragments. The complementary bases of the new DNA are synthesized onto the parental DNA by a DNA polymerase III, starting from the location of the primers. The synthesis of the lagging strand is initiated by multiple RNA primers, as opposed to the leading strand, which only requires one primer. DNA polymerase I then removes the RNA primers, filling in the gaps made by the primers with the appropriate nucleotides. For the lagging strand, there is an extra step where the enzyme DNA ligase uses phosphodiester bonds to seal the gaps between the Okazaki fragments. Upon the completion of the DNA synthesis, DNA polymerase I and III comb through the two new daughter strands, looking for and repairing any incorrect base pairings.